Western blotting procedure, proteins are first isolated from the rest of the cell's components. To extract proteins from cells, researchers use chemical-based and/or physical-based methods to disrupt the cell's plasma membrane and release the proteins inside it. Chemical-based lysis uses various types of detergents to make proteins accessible. For example, weak detergents are used to extract water-soluble proteins inside the cell without disrupting intracellular membrane compartments. Strong detergents are able to solubilize or make available the proteins found within all membranes. Following lysis, cells and their contents are spun in a microcentrifuge. This results in an aqueous supernatant, which contains solubilized proteins, and a pellet, which contains membranes, organelles, nucleic acids, and any remaining insolubilized proteins. The supernatant, now called a protein lysate, is separated from the pellet to use for Western blotting. The most common type of protein lysate that researchers prepare is called a whole cell lysate because it contains all of the proteins within a cell. Two other commonly prepared types of lysates are nuclear and cytoplasmic lysates, which contain only nuclear and cytoplasmic proteins respectively, often prepared to study the regulation of protein localization to these two compartments. A fourth commonly used lysate type is an immunoproteins are loaded onto a gel and separated from one another using an electrical current during polyacrylamide gel electro according to size, shape, and/or charge. To accomplish this, protein lysates are loaded onto a gel matrix. The matrix is made up of holes through which proteins can pass. The smaller the protein, the faster it will pass through the holes in the gel matrix. The bigger the protein, the longer it will take to go through the gel matrix. To form a gel matrix, a catalyst and polymerizing agent are added to a liquid solution containing two different acrylamide compounds, acrylamide and bisacrylamide. The catalyst and polymerizing proteins are assessed in a denatured or unfolded state and separated in the gel matrix according to their mass only. To accomplish this, sodium dodecyl sulfate, or SDS, an anionic detergent, is added to denatured proteins and uniformly coat them with a negative charge. This means that proteins with similar mass but different shapes migrate at similar rates in an SDS page. By the same token, proteins with similar mass but different overall charges also migrate through the gel matrix at comparable rates. This property of SDS page makes it particularly suitable for assessing changes that affect the mass of a protein, such as covalent modifications or changes in protein expression. During SDS page, SDS is added to the gel solution, protein samples, and running buffer to ensure that proteins remain negatively charged and unfolded throughout the procedure. For the rest of this animation, we will use SDS page to illustrate subsequent steps in the Western blotting procedure. Before running a polyacrylamide gel, researchers need a way to easily load the protein samples into the wells, which are immersed in running buffer, and monitor the proteins as they migrate through the proteins and/or molecules. The proteins are transferred from the gel onto a more durable material that can be manipulated and blotted with antibodies of interest. Transferring is necessary to allow antibodies free access to proteins and because polyacrylamide gels are fragile and difficult to manipulate. The transfer method most commonly used in Western blotting is electroelution, where an electric field is applied to the gel to facilitate the migration of the proteins out of the gel and onto a piece of membrane. Transfer is accomplished by placing the polyacrylamide gel in direct contact with a membrane made of nitrocellulose or another non-reactive material that binds to proteins and submerging both in a buffer-filled chamber. The gel membrane sandwich is placed in such a way that the negative electrode is located closest to the gel, while the positive electrode is located closest to the membrane. When the electric field is applied to the chamber, proteins in the gel migrate from the negative end to the positive end of the chamber, moving out of the gel and into the membrane, to which they become tightly attached. In the next step, the membrane is probed with an antibody that recognizes the protein of interest, specific residues, or chemical modifications. The membranes used for Western blotting have a high affinity for proteins, including antibodies. Therefore, before antibodies are added to the membrane, it is important to minimize nonspecific antibody binding to the membrane. The membrane is incubated with a solution typically containing a blocking agent, such as dry milk or purified proteins, in a mild detergent. Proteins within the blocking agent will bind to the areas within the membrane that are not already occupied by bound proteins, preventing nonspecific binding of antibodies in subsequent steps. After blocking, the membrane is then incubated in a solution containing blocking solution and the primary antibody that specifically recognizes the protein of interest. Incubation is then followed by a series of washes with washing buffer under mild agitation. Washing buffer contains the same mild detergent used in the blocking solution, which helps remove nonspecifically bound primary antibody. After washing the membrane to remove unbound primary antibody, the membrane is incubated with a secondary antibody, which recognizes the type of primary antibody used. As with the primary antibody incubation, incubation with a secondary antibody is performed in the presence of blocking solution, followed by a series of washes. Secondary antibodies are bound to a reporter enzyme or a fluorophore, which allows for the visual detection of primary antibody binding to the protein or modification of interest. Secondary antibodies also amplify visual detection because multiple secondary antibodies can bind to a single primary antibody. The most commonly used secondary antibody type for Western blotting is conjugated to the enzyme horseradish peroxidase. Horseradish peroxidase catalyzes a reaction that produces light as a byproduct. The amount of light produced is directly proportional to the amount of horseradish peroxidase conjugated antibody bound. The light signal is a transient product and will only persist while the reaction is occurring. The most common way to record light output is using X-ray film. The X-ray film is placed directly on top of the membrane after the addition of the horseradish peroxidase substrate. Increased time exposure of the membrane to the film will yield stronger